Moving to real data sets. So we will start using one of the most common technologies, which is Visium. So probably you are already familiar with this technology, but if not, just as a real brief introduction to how this technology works, basically the idea is that in your sample, you have kind of a grid arrangement, which is basically an arrangement of circles or spots. But this technology doesn't have the single cell resolution. So here, each spot, let's say this is your tissue here, where you can also capture the H&E, like coloring your tissue, or even use some immunofluorescence for protein detection. So you can capture the tissue image or immunofluorescence, but also you will get a whole transcriptome detection in each spot. Now, each of these spots is basically just in an arrangement of circles, but each of these circles, something like similar to this image in the right, each of these circles will contain all transcriptome information, but will cover several cells from your original sample. So just keep in mind that sometimes I refer to each of these spots as cells, and even in the Giotto framework, we have kind of a cells by gene matrix, and we usually call these individual spots as cells. But let's just keep in mind that they are not single cell level. There are other technologies that we will review during the workshop that they are single cell technology. Just keep in mind that in this case, each spot is covering around 6 to 7 cells. But we are still able to get a good sense of what is happening in our sample. So for this exercise, we are going to use a sample coming from mouse brain tissue. This is a publicly available data set on 10x Genomics, which is the company that is able to sequence the Visium samples. So you can find it directly on the website of 10x Genomics, following the links that we provided in the book. You can also get more information about for example, the patient that provided the sample, the sex, age, etc. If you haven't downloaded this data set yet,
Awesome. Can you make the font a little bit bigger? Command, yeah,
this font?
perfect. Yes.
Yes. Okay.
Perfect. Thank you.
Yeah. So if you haven't downloaded the data set yet, you can follow also these instructions. So you can either download it directly here following the link to the original website, or we have copied already the link for download it using just the R commands. So I invite you to create a specific folder in your current session, in your R session, called data. And then we are organizing the data sets according to the name of the sessions. So in this case, this session corresponds to the day 1, and we are on the session number 5. Just to have a little bit of organization in case you want to follow the same structure. So from there, basically, you can use this command, download file. You need to provide the full link, and then provide also the name for the destination file. So you here need to provide also the structure, like the data folder, and then the session subfolder, and then finally a name for your sample. Now you will need to download 2 files. One is the feature matrix, which is containing basically the reads in an arrangement of genes in the rows and cells or spots, in this case, in the columns. And then you will have another file that is the spatial one, which contains basically a table with the spatial locations, the X and Y location for each spot. Both of these files are compressed when you download them. So we have also provided the instructions for unzip them, for uncompress them. or if you are in a Mac computer, I think it also works if you just double click on your file. So you can do any of those. You can follow any of those options. So once that you uncompress these zip files, you should get two folders. So you should get the raw feature VC matrix and the spatial folders. So let me show you how this should look like in your folders. So let's say that you are here in your main station and you go to the data folder, then for the session 1, sorry, for the day 1, session 5, you will get first the unzipped files, something like this. And then once that you uncompress them, you should get these two folders, raw feature VC matrix and spatial matrix and spatial folder, sorry. So inside raw feature VC matrix, you will have this type of arrangement. So you have three files, barcodes, features, and then the matrix, which contains basically the grids. And then in the spatial folder, you have the images of the tissue, and you have a high resolution image and a low resolution image. So in the low resolution image, basically it's showing the image of the H&E image of the tissue. Usually we load this one for the analysis because it's a very light image, but if you want to create very nice plots at some point, you can also indicate to read the high resolution image instead of the low resolution one. So all of this information is contained in these two folders. Now you don't need to worry a lot about how to read the individual files because we already provide in Giotto one function that helps reading everything together. So let's go to the code. Either if you are following the book or if you have access to the RMD. So if you are in the book, you can copy each section of the code going to this upper right corner in each box with code. So basically you can click here and then copy this well, it will copy the code and then you can paste it in your R script. Or you can also go to the GitHub repository where this book lives, and you can download the R Markdown files. So the first thing that we need to do is, of course, loading Giotto. Now, if you were following the code from the previous session with George in the morning, what I suggest is to restart the session so that we have a clean environment here. So you can do this just going here to the top menu, session, and then restart R. And once that it's done, you will see your environment completely clean. Let me make this bigger. Okay. So we need to load Giotto again, of course. Since we restarted the session, then we need to load it again. So as George was describing in the morning, this is the umbrella package, and internally it's calling for the modules of Giotto. In this case, it's calling for Giotto class, for example, because it's one of the basic modules that we require for building the Giotto object. Now, we are going to set some instructions inside of the Giotto object. For example, we need to indicate where we want to store our results. So in the same way as we organize the data folder and then we are providing the number of the session, I would suggest that you also follow this structure so that you can have separate the results from today to the results from tomorrow from the other technologies that we are going to review. So I'm going to set the results folder as results and then under the subfolder day 1, session 5. Then you also need to provide the Python path. As you probably remember it from the morning, this package is mainly written in R, but we are also calling for a few Python modules for very specific task. So we need to provide the path where our installation on Python is living in our computer. Now, if you don't remember where your Python is installed, you don't have to worry about it, because if you set the Python path as null, then the function to create the Giotto object and to create the instructions will look automatically for any available Python path. But if you have several installations, or for example, several environments, several content environments, then probably you want to provide here manually the path to your environment. By default, the functions will look for the Giotto environment in Miniconda that you created using the install Giotto environment with the function. So if you just set this to null, it will automatically look for the path. And then we will use the create Giotto instructions function, basically to setting additional things. Here we are going to basically pass the results folder path that we just created above here. We are also passing the Python path. If you don't want to create individual variables, you can also directly here type the paths. And also something important is to let know the instructions of Giotto whether you want to save, show, or return your plots. You can set everything to true, or you can just leave one of them to true. In this case, for example, I'm saving the plot, but I'm not showing them. I could change this to true, for example, if I want to also show the plots at the same time as I'm saving them in the folder. So let's create instructions. We can also print the content of this variable if you want to make sure what you just created. So this is basically a list of parameters. In this case, it's automatically detecting my Python path. For example, here we have the message saying that a Giotto Python environment was found, and it's showing the path to that. Then it's showing the show plot equals to true, the return plot will be false, then it will save my plots. It's showing where I'm going to save my plots. The plot format is something that you can also change, but the automatic or the default value is saving the plots as PNG files. Then you have also the resolution for the plots. This is something that the function create Giotto instructions is creating by default. But if you want to modify the values, you basically just have to add the other arguments here after this list of arguments that I provided. So also some additional details for the plots, like the units, height, width, and at the end, it's just giving me the class of this object. Okay, so then I also need to provide where my data is living. So if you follow the recommended structure for downloading your data set, you will have your data stored in this path, which is data and then the one section 5. If you created a different a different folder with another name, or it lives in a different path in your computer, then you need to modify this path here. In my case, I have them under data in the one section 5. Okay, finally, we are going to create the Giotto object. So I was mentioning before that in this data folder, we have 2 main directories, and then inside of them, we have several files, but that we don't need to worry about how to read the individual files, because we have this very nice function, which is going to look on inside of your directory, and it will look for the standardized organization of the files. So having this subfolder, raw feature busy matrix, and the spatial one is something that happens for all the data sets in Visium technology. So independently of what sample you are downloading, or if you are sequencing your own sample, the company will send you several files, but the 2 more important ones are the zip files that we just downloaded with the raw feature matrix and the spatial information. So at the end, you will always end with these uncompressed folders, which is the feature VC matrix and the spatial folder. So since it's a standardized organization of files, then this function will look for this structure and it will automatically read all the needed files here. And it will create the Giotto object based on that. So basically the important arguments here is providing where these subfolders live. So you don't need to provide the path to the specific VC matrix or the spatial. The important thing is that you place these two folders in the same place. In this case, both of them live in the data path that it just created above here in this section 5, sorry. Then the default is that we are going to look for the row counts, because if you go to the dataset website, you will see that there is also a folder or a file that it's called filtered feature matrix. In this case, we are downloading the raw feature matrix because we want to do the processing of this file by ourselves. But in case you want to use the filtered matrix, then you need to change here the name. Instead of raw, you need to establish filtered as the value in this argument. And then here is where you can change whether you want to read the low resolution image, which is the default, or if you want to read the high resolution image, then you just need to change the name here of this file and provide the right name for the high resolution image that you have in the special folder. And we are also passing the instructions for creating the Giotto object. Okay, so if we run this function, sorry, it will take just a few seconds. So it's saying that the image was found, and there are also there is also some information living here in Spatial regarding the scaling for the image, so that we can align the spot coordinates to the size of the image. But that's information that we are not creating by ourselves. That's information that lives here also in the Spatial folder. So once that we created the Giotto object, we can even print it, and it will just give us a summary, kind of just showing what we have. For example, saying that we have a spatial unit, which is the cell level. In case you are dealing with a subcellular data set, you will see this in tomorrow, I believe, or in the third day. If you are dealing with subcellular data set, then you will see that another spatial unit will be created for the individual polygons, for example. But in this case, since we are doing things not in the subcellular level, then the only spatial unit that we have right now is the cell level. Now just remember that in this case, Visium is handling the spots. So even if we call them cells, just keep in mind that they are spots in this case. And then the feature type, in this case, the default is RNA. But if you are dealing with proteins, which you will see in another session, then you can change the value here. And instead of saying RNA, it will say protein. From there, since we don't have subcellular information, it's basically just saying that the polygon is the same as the basic unit, as the general unit, which is just cell. And then in the expression level, is saying that we have only the row counts. The Giotto object is able to store the different results after performing some steps. So right now, we haven't performed any additional processing on the expression matrix. So the only thing that we have right now under the expressions slot is just the row counts. But you will see as we go in the steps of this analysis that more things will start appearing here after the row section. For example, the normalized counts or the scale counts when we perform those analyses. And then regarding the spatial locations, we only have the row spatial locations. In this case, this will not change because basically we just will be operating under the same level. Let's say we are not going to do any modifications on the spatial locations. And then in the images, it's basically just listing that we have one image. But keep an eye on this slot when you go to the section of handling multiple samples, because then you will be able to see changes here under the image section. Right now, we are only handling one image because we only have one sample. So that section is not going to change during this analysis. Okay, so something that we can do now is evaluate or visualize what spots in our sample are aligning to the tissue and which ones are not. So we can visualize this using the spatplot2D function. Basically, this will create a spatial plot, and then we need to provide, of course, the Giotto object that we are using here. And we want to color the cells depending on the information of the metadata regarding the in-tissue. So this is information that comes also attached to the dataset. We are not creating this column manually. It's already coming with the dataset. And we can also use the metadata, the pdata.dt, to get the metadata, the cell metadata table. So let's do that first instead of going directly to the visualization. So let's do the Visium brain. And here we have just a few lines from our metadata table. So you can see that the information contained is the cell ID. Then we have the column already called in tissue showing 0 or 1. The 0 indicates that this cell or spot is not overlapping with the tissue, while the value 1 is saying that this cell or spot is overlapping with the tissue. And then it's just showing basic information regarding the rows and columns where these spots are located. So basically, we are using this column to color our spatial plot. Then I'm passing also a particular point size to just make them smaller or bigger. You can modify this depending on how you want the visualization to look. And in this case, I'm also coloring with a specific colors my spots. So I'm passing this vector where I'm indicating from the in-tissue column, I want the value zeros to show as a light gray color, while the ones, the spots with a 1 in this column, I want them to show in blue. And I also want to show the tissue image. So that's what I'm indicating with these arguments. Okay, here I have my plot. Okay, so I think this is looking good. So basically what I'm observing here is that I have as I was saying, in blue, just the spots that are matching with the tissue, while the spots in gray are basically indicating that those spots don't align with the tissue. Now, probably since the spots are kind of big, they are covering most of the tissue image. But if I make them a little bit smaller, or if I also modify the transparency, then we can observe better the tissue on the back. So here, when I am making them smaller, then we can observe in the background the tissue image. So that this can also help you just to verify that the metadata is actually corresponding very well with the location of your sample. So in this case, yeah, they look like the blue is actually corresponding to the tissue correctly. So we are going to use now that information in the metadata table to subset the spots. And we only want to keep those that are aligning with the tissue, right? So we want to get rid of all the zeros, basically, of everything that in the metadata table is marked as 0. We want to get rid of them, and we want to keep the ones. So let's use that information to filter our sample. So you can either use the pdata.dt as I used before and passing the name of your Giotto object. This will give you automatically a data table. But we also have the function get cell metadata. And as the output, the default output would be a particular object, which is a metadata object. But you can also indicate in the output argument and set this as data table. So you will get exactly the same result, which is just a metadata table. We can verify here, just printing this metadata value. So you can see that it's exactly the same result. So here I'm just extracting the information that is already in the Giotto object to do some filtering. So from this metadata table, what I want to do is basically keep only the rows where the in tissue value is equals to 1. So basically is what I have in this row, in this code here. So I'm just filtering in tissue equals to 1. And then from the resulting table, I want to keep the cell IDs, right? So I want to know which cells correspond to the in tissue barcode. Once that I have the list of cell IDs, let's keep an eye let's just see the head of this vector. This is basically just a vector with cell IDs that correspond already to the in tissue equals to 1. And I'm going to use this vector. This is a very long vector. If we take a look at the length of in tissue barcodes, so we are having 2,700 with 2 cells corresponding to the in tissue section. And then we are going to use this function called subset Giotto. So subset Giotto will basically use cell IDs. That's basically what we have right now as the filtering, let's say. So for subsetting, we need to provide all the cell IDs that we want to keep. And then it will go through the different slots and will automatically also subset the other slots. Right now, we don't have additional information. For example, we don't have the PCA yet or the UMAP. But in case you already had that information, then subset Giotto will automatically also subset the tables, like the other slots, like for example, the PCA table or the UMAP table, and will only keep these cells that you are providing in the cell IDs. So it automatically checks all the slots and it subsets everything. So let's pass here our in tissue cells. And if we observe now the Visium brain again, basically I have right now the same information, the same summary as before. I still have the same spatial unit. I still have the same feature type, which is RNA. Then I still have the row counts and the image, right? The image is not going to be modified. Basically it's just the same file. The only thing is going to be modified in this case is the spatial locations table and also the expression information with the corresponding metadata. Okay, so if we take a look again, let's say that I want to just get again the metadata and we print this, you will observe that now we only have ones in our in tissue column here, right? So we got rid of the zeros. All right. So moving on to the quality control. We can create a count of how many features we have in each of these cells or spots, meaning that we want to know how many genes from the whole transcriptome we were able to capture in each of these spots. So we have a function in Giotto called add statistics. Will basically perform. It will perform automatically that counting. So it will evaluate for each of these cells how many genes from the whole available genes in this data set, how many genes it was able to capture per cell, and what is the percentage also of these what is the percentage of features per cell based on the total availability of genes in this sample. So I'm going to create just a temporal Giotto object that will only store the statistics, because I don't want to keep those statistics for the future. Not right now. I just want to kind of test how good is this sample. So I will basically output I will redirect the output of the other statistics to a new Giotto object. And then out here, important is to indicate in the expression values argument that we are using the raw counts, because we don't have other type of counts right now. And the default value for this function is using the normalized counts. So if you are doing the quality control test, then you need to specify that you want to use the raw counts. Okay. So let's take a look at the Visium brain statistics object. We are going to use again the spatial plot function, but this time instead of coloring the cells by the in tissue column, we want to use a new column available now in the metadata. So let's take a look now at the cell metadata of this object. So let's observe that we have new columns. Before we only had until this column, the array O, but now we have additional information. Now we have number of features, which is basically indicating the number of features per cell. It's this number. Basically, this is the number of features in this cell. Then this is the number of features in this cell, and so on. Then what's the percentage of features? Basically, from the whole transcript of in this sample, how much is what's the percentage of this value? And the last thing is that we have the total number of counts in this cell, independently of the number of genes. We just have the total expression as in total number of reads in this cell. So we are going to use this column, number of features, to color our cells. And in this case, this is going to be a continuous scale, right? So we don't want to treat these as different and individual groups, as in the case of 0 or 1. In this case, these will categorical values. And in the case of the number of features, we want a continuous scale. So we need to set the argument color as factor equals to false, so that we can treat the colored scale as a continuous scale. And this is how it looks like. So basically showing every cell in or every spot in this case in our sample, and then in the color scale is showing how many features I have per cell. So in this case, we can observe that the maximal and minimal value, and from here we can start making decisions on, for example, what value should I use to remove the cells that contain a very, very low value here in the number of genes per cell. And it's also nice to observe this plot just to have an idea on how much of our tissue is containing spots with a high number of features per cell, or whether, for example, the majority of our sample is looking in the deep blue, then we probably have to take up in consideration those values, or probably even make the decision of whether this sample is completely bad, for example. In this case, this is a good sample, so we have good values on these spots over here. Now, since it's difficult to see specific values here to establish a threshold of what is the minimum number of features that we want per cell to keep those cells, then we need to use additional plots. So let's create another plot, which is called filter distributions. We are going to use this function, filter distributions, and this will create a histogram that shows basically the same information, the number of features per cell. But of course, it's easier to see the information as a distribution instead of just the color scale here on the spatial plot. So basically, when we are providing here in the argument, what is the type of detection that we want to perform? If we establish a detection equals to cells, we are indicating that we want to create the histogram based on the number of features detected per cell. We will see in a minute what's the other option if we change the value of that argument. So here we can observe basically the distributions. We can see that most of the cells have around 6,000 to 7,000 of features per cell, which is a really good number. Then we have basically the distribution to the other sides. And we can see that we still have some cells over here that have a low number of features per cell. So probably we want to get rid of these cells because those are not going to provide a lot of information. And we probably just want to keep the cells in this big area. So, okay, this is a histogram of features detected per cell. I particularly find this the most useful, but we also have the option to create a different histogram, which is plotting the number of cells with a certain number of features. So if we change the value here in the argument and we set up as features, as fits, basically meaning the features, then let's observe what's the result here in this histogram. So in this case, instead of plotting the number of features per cell, we are plotting the distribution of the features detected in a certain number of cells. So they are kind of related, but they are giving us basically different information. So instead of plotting in the X the number of features, and as the counts, the number of cells that contain that amount of features. Here we are saying, okay, how many cells contain a particular number of features? So for example, the way to read this is that if we observe this bar over here, basically we are saying, okay, how many cells in my data set have around 1,000 of features per cell? And basically, this is the number of counts of cells with that particular number of features. And what we have to probably what we want to get rid of in this case is this big bar, which is containing the cells with 0 fits or 0 features per cell. So basically here, we are observing that we have a high amount of cells. Probably this is not a very, very specific range. And here we are observing around from 0 to 100. So we are, in this bar, we are capturing the number of cells that contain around from 0 to around 100 of features per cell. So that's why I say that I prefer either histogram, because usually I found it easier to interpret. But basically, that's how you interpret this one. So we want to get rid of this bar, basically. We want to get rid of this count, which is containing the cells with a very low number of features. That's the main idea. Now, how do we know what's the best value to set as the features, as the minimal number of features per cell, and also the minimal number of cells that I want to a specific containing some of these genes? You can basically, one option is that you can run the filtering step, which is coming in the next box. Running multiple times this filtering Giotto and play a little bit with these values, which are the most important ones. The features that are detected in a minimum number of cells. In this case, I'm setting this to 50, or the minimal number of detected features per cell, which I'm setting as 1,000. You could manually play a little bit with these two values and see what's the best combination. But we also have a function that helps performing those tests without manually changing the values. So filter combinations is basically for providing different values in the most popular filtering arguments, which is the features detected in a minimal number of cells and the minimal detected features per cell. And then you can also play with the expression threshold. So in this case, I'm providing 3 different expression thresholds, and then I'm providing 3 different values for the minimal cells and the minimal features per cell. So the output of this function is a plot that shows you how many cells and features you will remove from your dataset with each combination. And it's grouping them, in this case, by the threshold. So for the threshold with a value of 1, we have the combination of removing or setting as the minimal number of cells with features equals to 50. So I want that each gene on my dataset appears in at least 50 cells of my dataset. That's the how to interpret this value. And I'm combining that argument or that the value of that argument with also asking that each cell should contain at least 500 genes. If I use this combination of arguments, like 50, 500 with the threshold equals to 1, then I will remove around, I don't know, probably like 25 cells. In the X is appearing the number of removed cells with this combination. And I will be removing around like 7,000 of genes or so. Yeah, like 7,000. So with that combination, that's the number of features and cells that I will remove. Then if I look at the next combination of values, basically I'm still using the threshold equals to 1. But then I'm saying I want that each gene appears in at least 100 cells in my dataset. And I want that each cell contains at least 1,000 of genes. With that combination, I'm losing or I'm removing around the same number of cells, but I will remove more genes. And then if I go to the next combination, I will have still the threshold 1, but I'm saying, okay, I want to have at least 200 cells with, I want each gene to be expressed in at least 200 of cells, but I want each cell to have 1,500 of genes per cell. So with that combination, I will remove around 10,000 of features and probably around 40 cells or so. So basically, you can take each of these combinations that you have provided and from here make a decision on what's the best value to use for your filtering step. So in green, we have also the same combination in these values, but with a different threshold. In this case, is using an expression threshold of 2. And then with the blue ones, we have an expression threshold of 3. So of course, if we increase the expression threshold, we will get rid of more cells and more genes. So in this case, with the most astringent value, we will removing more than 15,000 of genes. So that's something that we don't want to do. We still want to keep as much information as possible. So for this data set, the values that I picked was using the expression threshold of 1, then asking for each gene to be expressed in at least 50 cells from the whole data set. And then I want that each cell contains at least 1,000 of genes. And what expression matrix I want to use for applying this filtering, where I'm using the raw values right now. Okay, so basically I want to take the raw counts and apply these filters on top of that raw matrix. And then I'm just setting the variables equals to true so that it gives me all the information about what's happening in this step. So here, it's basically saying, okay, it's subsetting the expression data. So it's removing all the genes that don't accomplish with this threshold that I set. It's also removing some cells. So since it's removing cells, we need to update the spatial locations because we don't want the coordinates of the cells that are not going to be in this data set anymore. We also need to update the cell metadata table to do basically the same, to remove the information from those cells that will be removed in the filtering. We also need to update the feature metadata because in the feature metadata right now, what we have is the feature IDs or the gene IDs in this case. We don't have spatial networks or dimension reductions networks, but since you can perform this step, even if you already perform other processing steps in your object, so it will check just to make sure that if it needs to perform some subsetting or not. So at the end, it's also showing how many cells and features were removed using this combination of settings. In this case, we are only removing 4 cells from the original number of around 2,700 of cells. And then from the original amount of 22,000 or so of genes, we are removing around 7,000 of genes. Okay. Now that we have filtered the cells that don't have a good number of features, and those features that don't appear very frequently in our data set, now we need to perform the normalization so that we can make comparable the counts between the cells in our data set. So the scale factor here has a default value of 6,000, but you can play with it and choose the better for your data set. This is a number that works very well for this sample. So in this case, it's performing the scaling also automatically. So it's doing 2 steps. It's creating 2 outputs. The normalized counts, but also the scale counts. So in the case of the normalized one, it's just going from 0 to the top of the counts in our data set. But in the scale matrix, then we are putting as the middle number 0, and from there is going to positive values or to negative values in the case there are down expressed genes. So if we take a look at our object again, you remember that in the past, we only had in this slot of the expression the raw matrix. But now, after performing the normalization step, we have 2 more matrices in the expression slot. We have now the normalized matrix and the scale matrix. And these are matrices that we are going to use in the next steps. So once that we have performed the normalization, probably we'll want to calculate again the statistics. So this time the statistics function is working on the normalized counts. So it's this time the metadata table and the column where it's showing the number of features those number of features will be the result, or will be a little bit different than if you take into account the raw number of features, because this time we remove all of those features that didn't accomplish with the filtering thresholds. So if we take a look again at the metadata of the Visium brain, we still have the same columns, the number of features, the percentage of features, and the total expression. But this time, this number of features, it's only considering the features that accomplish with our filtering thresholds. And the percentage of features, of course, will change a little bit. because the total number of genes that we have now in the dataset is different than the original. And finally, in the total expression, you can observe that this time we don't have integers anymore, because in the past, when we performed the statistics, we were counting the total number of counts in the raw matrix, which has only integer values. In this case, is considering the normalized values, which have also decimals. So that's why this is not integer anymore. So this total expression is normalized expression. If we want to visualize this again, then you can basically take the spatial plot function and pass one more time in the cell color argument the number of features column, and just remember to set the color as factor equals to false so that we can get a continuous scale in the color. So let's take a look. And this is basically how it looks like. The distribution, the spatial distribution of the number of features per cell in our sample after doing the filtering. And you could even create also a plot using the total expression so that you can observe the distribution of the normalized counts. So let's do that. I did include this code in the book, but I can update it after this session. So just instead of the number of features, I'm going to put the total expression. So basically here we are visualizing the normalized expression distribution in our plot. And looks something like this. So here you can observe that we have deep blue colors, and then we have up to the top of the normalized expression. Okay, right. So after we perform the normalization, we probably want to evaluate whether we have highly variable genes in this data set. Because remember that right now we have around 15,000 of genes in this sample. So we had originally around 22,000, then we remove about 7,000 in the filtering step. So that means that we have around 15,000 of genes still here. So handling the 15,000 of genes may not be the best for computational resources. So we probably want to just find those values that have some variability across the sample. So since there are some genes that will have, for example, the keep housing genes, those will have around the same expression in the different cells. So we are only interested on the genes that have some change in the cells from our sample. So we are going to calculate the highly variable features, and we have 3 methods available, depending on what's the behavior of the expression in your sample. So we have the lowest regression method, and in this one, basically, you want to use this method when you already know what's the relationship between the expression and the variance of your sample. So if you know that this relationship is not linear, and it can be basically described as a non-parametric model, then you can use this method for finding the highly variable features. And the output of having this calculation will look something like this. So it's creating the plot. Let me see. I think I didn't see there. I can look in the results. Okay, so it should look something like this, where you can observe exactly what I was describing. The relationship is not linear, so it's more like a logarithm relationship between the expression and the variance of the gene expression in your data set. And what we are highlighting here in yellow is those genes that are identified as highly variable features. right? And in gray is basically those genes that didn't have a really high variance, sorry, a really high variance in your data set, okay? Then the other method is the Pearson residuals. So in this case, you can use this method when you know that there are groups of genes over dispersed. And you want to basically highlight those genes and identify them as the highly variable features. Now let me see what happened here. Okay, for some reason it's showing an error. I'm not completely sure why this happened, but oh, I see. I think it's something here. Let me change this in options. I hope that this works. Let's try again. Okay, yes, there we go. So in case you get the same error, it's just regarding some options from R regarding referring to the maximal space that some functions can occupy. So you can basically just set or change the options here and establish as infinite value so that you don't have a limit. Okay, so this plot was generated. So the output in this case looks like this. As you can observe in this data set, we don't have really a good amount of genes that have this property of showing a really, really high variance. So the number of genes that we are identifying as highly variable features is very small. So this is not the best method for this data set, but I just wanted to show you the methods that we have available in Giotto. But of course, the selection of the best method is up to you, and it depends in the characteristics of your data set. So as you probably know, there is not like a exact recipe for analyzing all the data sets. You need to consider the technology that you are using. You need to consider the sample source, because also depending on the tissue, you could get best number of genes per cell or per spot. There are other tissues that are more difficult to handle. So yeah, the purpose of this workshop is showing what we can do with the Giotto package, but of course, choosing the best arguments to use and the best values for those arguments depends a lot on each case. So just for this method, basically the idea is that if you have genes with a really, really high variance and you want to highlight those and select them as the highly variable features, then you can use the Pearson's residuals method. Then there is a third method, which is the covariance groups. This is the default for this function. So if you don't set or you don't specify the method and you just provide the Giotto object as the input for the calculate HBF, this is the method that will be used. So the covariance groups is basically when you don't know the relationships between the expression of your data set and the variance. So you cannot make sure that there is a nonlinear relationship, as in the first example, in the lowest method. And at the same time, you are not sure if there are specific genes with a super high variability. So instead of highlighting genes with a really extreme variance or trying to find a nonlinear relationship, what this method will do is that it will create different groups and will try to highlight the genes that have a high variance in each group. So let's see how this plot looks. So basically, it's grouping genes depending on their behavior, depending on their variance, which could correspond with, for example, different cell types of different regions on your tissue. So it's basically grouping them by their behavior, by their variance, basically, and then it's highlighting those genes with a highly variability, with a high variability in each group. So from each group, it will select the genes with the highest variability, and it will label them as the highly variable features. And basically, at the end, we want to keep only these genes in yellow. So that's how, in general, how the method works. So I'm going to keep basically the result of this method, of the latest one, and using those highly variable features, we can go to the next step of the analysis, which is the dimension reduction. So probably you are already familiar with the principal components analysis, but just for those who are not very familiar, basically this components analysis is trying to make a combination of genes and try to identify which combinations of genes are explaining better the behavior of our sample. So instead of handling hundreds of genes or thousands of genes, and then we can refer to the combination of the genes that explain the best our data set and just treat them as an individual component. So we will reduce from having hundreds of genes to just have one component, which is the combination of them. Now, of course, we don't want to calculate just one component. We want to calculate multiple combinations of genes. So the default value for running the PCA is that we will calculate the first 100 components, but of course you can modify this value. So if you go to the help of run PCA, you will see this argument NCP, which is the number of principal components. As you can see here, the default value is 100, but you can play with them. So in this case, run PCA will also automatically look for the previous calculation of the highly variable features. If it finds, if the code inside of run PCA finds the previous identification of highly variable features in your sample, then it will take those highly variable features to calculate the PCA. That will be an automatic thing. But if you want to use all the genes in your sample or all the features, not necessarily genes, but for example, proteins, all the proteins in your sample, then you have two options. You either don't calculate the highly variable features, so basically you skip the previous step, or you can also set to null the features to use here. Because as you can see, it's, the default is looking for highly variable features as the features to use. So you can also set this to null or even fill it with all, and basically it will take into account all the features available in your dataset. In this case, we want to consider the highly variable features that we calculated before to run the PCA. Now, where is this column living? You may wonder that. Where is this HBF column? Okay, probably you remember that I mentioned that as in the same way as we have cell metadata, we also have features metadata. So we can use the accessor function, which is get feature metadata from our object. So we need to provide the Giotto object. And here you will get the information of the features. In this case, we have genes, but if you have other type of features like proteins, then you will have the information here in the same place. So it's giving us the information about the spatial unit, then the feature type, and in this case, it's basically the spatial unit is cell, then our feature type is RNA, and from there it's giving us the feature ID. So here we have the IDs from all the genes in our sample. Then it's showing also some statistics. This was added when we run the add statistics function. So it's showing how many, it's showing in how many cells this feature was detected. So for example, this gene is detected in 251 cells from our data set. And then it's also giving us some percentage of the cells, the total expression from this gene in our sample, the mean expression, etc. And at the end, we have one additional column, which is HbF. So basically, those genes that are detected as being highly variable features will be labeled as a yes, and those who are not a highly variable feature have the label as no. Here we are only seeing the first three features, but of course, if you print the whole table, then you can see all the genes in your dataset. So going back to the PCA. So basically, it automatically looked for the existence or not of this column in the feature metadata table. Since in this case it found the column, then it took this information into account and only took the genes who are highly variable features to calculate the 100 PCs. You have also another option for calculating the PCA, which is providing a specific list of features. This is very useful, for example, when you calculated before the existence of genes with spatial patterns. So if you are able to calculate features or genes who show a specific distribution on the spatial context of your sample, then you can subset them and calculate the PCA based only on the spatial genes. We are going to see the calculation of spatial genes tomorrow. So right now, we don't have that information because we haven't calculated them, but tomorrow we will have that. So we will rerun this step tomorrow. But right now, just to show you how this works, basically, if you have any list of features and you want to use that feature to those features to calculate the PCA, then you can pass the IDs here as a vector. And then I suggest that you label this result with a different name so that you don't overwrite the previous calculation of the regular PCA. So here I'm just getting kind of a random list because it's just the head of the IDs, of the feature IDs in my sample. So they don't have a particular biological meaning at this point. I just wanted to show you how to pass the customized features for the calculation. So once that we run the PCA, we can create a script plot. In this case, I'm using just the first 30 components, not the 100, but of course you can use the 100 if you want. And what it's describing, probably you are already familiar with this, but if not, what this script plot describes is the percentage of variance explained by each of the principal components. So what you expect is to see that the first and second or third component explain the most of the behavior of your data set. So you expect them to have a good percentage of variance here. Now, we will see that, of course, as we move to the calculation of the other components, this percentage of variance will be lower and lower, but it's still contributing to the whole explanation of our data set. So in the bottom, we are showing just the cumulative of this percentage of variance. So yeah, as you can imagine, it's just basically showing the first component plus the second component, and then we have the result in this one, in this bar. And then the adding of the first three components, and then we have this bar as a result. Of course, it's good to take a look at this plot so that you can also evaluate whether the calculation of the PCA was good or not, or whether the number of genes or the features that you are using for this step is a good group or not. The next thing that we want to do is also plotting the components. So we are only going to plot the first and second component to observe the behavior of our dataset. Basically, we are just showing here the PCA values per cell. So each of these points is a cell, or in this case, it is a spot. So it's just showing the values in the principal component 1 in X and the principal component 2 in Y. And it's just showing here also, in addition, what's the percentage of variance explained by the principal component 1 and 2. Now, if you want to plot the PCA that we calculated using our custom set of genes, you can pass the dimension reduction name of the PCA that you calculated in addition to the main one. So if you don't pass additional arguments to this function, it will look for the first result in the PCA, which is the automatic name of the PCA, which is just PCA basically. But if you modify the reduction name, which is basically the name that is assigned to the result of the PCA, then you can provide any name that you have created in the past. And in this case, we used as the custom PCA for just passing the first 1,000 of genes in our data set. And this is how it looks, the PCA here. Of course, this doesn't look as nice as the other one, but it's fine. It was just like an example to show how to create our custom PCs. After that, after calculating the PCA, now probably we want to represent the behavior of the principal components, but in a two-dimensional way. So we are going to calculate the UMAP. So we have also that step included in Giotto as the run UMAP function. And we calculated 100 PCAs, 100 principal components, but as we were observing in the script plot, when we move to the higher numbers in the principal components, we don't get a lot of additional information. So usually keeping the first 10 components is good enough to explain most of the variability in our dataset. But of course, if you want to modify here the dimensions to use, and you want to use, for example, the first 20 components or the first 30 components, you can modify those values in this argument. The default value is taking the first 10 components. So we are running our UMAP, and then we can plot it. So using plot UMAP again will look for the default name in the resulting from running the UMAP. But if you go to the help of run UMAP, you can find some additional arguments where you can pass a specific name for the result. So I believe if you use name here, if you change name and instead of saying null, you put a different name here, then you can create multiple UMAPs and save all of them in the same Giotto object. And then for plotting them, you can also call them for this name that you have assigned before. So if you don't provide anything, basically the automatic name is just UMAP. And then the plot UMAP function will basically also look for either that automatic UMAP name, or you need to specify which name you want to use. So if you look in the arguments, I don't have it set. I don't see it here, but basically you provide the name. You can also provide here. Oh, here it's name of the UMAP. So here you can basically provide a different custom UMAP name in case you created something which is not the default. If not, don't worry. If you use just the default values, then the plotting function will also look for that name. So just plotting it again. Here it is. So we are observing here now the behavior of the first 10 principal components, but projected in a two-dimensional plot, which is the UMAP. And we are start observing here some clustering even if we haven't calculated the clusters yet, we start observing here some separation of the cells. Now, of course, there is also the option to calculate a t-SNE. So for calculating the t-SNE, we also use as the default just the first 10 principal components, but again, you can modify this value. The arguments for run t-SNE and for run QMAP are very similar, so you can also customize the name of your t-SNE, and you can create multiple t-SNEs under different names if you want to. Then after calculating it, we can plot the t-SNE. Again, here it will look for the default name, but you can customize that name if you want to. So here we are just showing the t-SNE dimension 1 versus the t-SNE dimension 2. Again, here we can observe that we start having some clustering of the cells or the spots, which is very nice. It's showing that we are on the right way. So to actually calculate the clusters, we need to first calculate the nearest network. So we need to find a numeric way to describe the distance between each spot or cell in our data set. So that's why we need to calculate the nearest network. Again, here we are using the first 10 principal components as the default. You can modify that value. And then we are also establishing the number of nearest neighbors to for the calculation of this network. But this is also the default value, 15, but of course you can modify it. A lot of people use, for example, 20. So you can use the value that works better for your data set. Now, the default, we have two options for calculating the nearest network. One is calculating the shared nearest neighbors, and the other one is calculating a k nearest neighbors. So using the shared nearest neighbors is what we usually is the default for this function, because then we need this information for calculating the latent clusters, which is one of the most common method for calculating the clusterings. But if you prefer to calculate the k nearest neighbors, then you just need to specify that you want to use this method under the type of network. So the type has as the default value S and N, but if you want to use the K and N, then you just change it here. So I will show you just very quick here. So we have those two options. The first one and the default is the S and N. So let me run the nearest network using this K and N. So they are a good thing about the framework in Giotto is that if you calculate two different methods for the networks, they are not going to be overgrading. So they are being stored as a separate network under the same slot, which is the nearest network. So if we just evaluate the slot names of this Giotto object, you can observe all the compartments or slots available. So you can observe that here we have nearest network. And inside of the nearest network, we have both the results of these two methods. So from the shared nearest neighbors and the k nearest neighbors. So a really cool thing about Giotto is that you have the access to all of these results, to the different steps that you have performed in the analysis of your Giotto object, but also to the to multiple results from a same step if you modified something. In this case, the result of this network will be under a different name, which is basically the K and N. So then you can call that result, that specific result, in further steps of the analysis. So, okay, once that we have the nearest network, we are going to move to the latent clustering. Now, for performing the clustering, we also have two methods. One of the most popular one is the latent clustering, but let me just connect the computer. But we also have the vein cluster integrated in Giotto. So if we want to perform the latent cluster, we need to calculate the shared nearest neighbors because it's the type of network that this method will look for in the default arguments. But if you prefer to use the k-nearest network, you can also do that. You just need to specify the network to use here. So as you can see, the default is going to look for the shared nearest neighbors, but you can modify here and type k and n instead of the s and n to use the other network that you calculated. Then another important value is the resolution. So the default resolution for calculating the clustering is 1, but for some data sets, you create too many clusters and you are almost reaching the level where you are separating each spot or each cell as an individual cluster. And of course, that's something that we don't want. We want to group them. So if you observe, I guess my recommendation is to try first using the resolution equals to 1, which is the highest resolution. But if you observe that you have too many clusters, then you start lowering the resolution. In this case, for this data set, we have already played with a little bit with the resolution, and the best value that we found is 0.4. But when you are working with new data sets, of course, you will have to play and calculate different resolutions sometimes. So it's going to take a little bit. Let's just wait. Okay, so the latent cluster was calculated. And this information, these results of the latent cluster is going to be stored in the cell metadata table. So if we calculate again, well, not calculate, we extract the metadata using again bdata dt of our Giotto object, you will see that we have a new column, which is latent clusters. And basically it's assigning the cluster ID to each cell or spot. Since we have this new column in the metadata, probably you remember that we have been using one function for plotting the spatial distribution of things in the metadata. So we are going to plot that in a second. We can use basically the name of this column in metadata to color the cells in the different plots that we have created before. So you remember that a minute ago we plotted the PCA, but we were just observing blue points, which were basically just the general individual spots. But now we can add another argument in the function, and we can color the points depending on the cluster ID that we have created in our metadata. So you can observe here, both in the legend on the side and also on top of the plot, the IDs of the different clusters. If we go and plot now the UMAP, and we also show, we are passing the same argument here, the cell color, and we are using this column on the metadata, then we can see that now the UMAP also has that information. Instead of just observing blue points, now we have the colors of the individual clusters here. And now we can even confirm that our processing workflow and also the clustering was good, because you expect to see kind of separated individual clusters here. If you see everything just overlapping, basically the colors are mixed together, then probably something wrong happened either with the pre-processing of your sample or with the clustering. And yeah, you need to modify something in your pipeline. But right now, we are observing a good separation of the different clusters here. Right now, something that I forgot to mention. Here, I'm setting the argument of showing the network as false, but you can activate this argument. So if you want to actually observe how is the connection between your spots or your cells, then you can set this argument as true. So let's plot again the UMAP, but now in addition to color each spot with the Leiden cluster, we are also going to show the nearest network. So this also helps to try to understand the connections. Usually you expect to see more connections or a stronger network between the cells corresponding to the same cluster and weak connections between the spots that are corresponding to different clusters. So you can also add the nearest network to your plot. Then we can also plot the TSNE. So you remember that we also calculated the TSNE in addition to the UMAP. So again, we are passing here in the argument of the cell color, the Leiden cluster ID. So let's observe this in bigger. So this is the TSNE. We are observing also that the clustering separation looks very good. So we see this is very clear, one separated cluster. We see this other cluster, the red one, the pink one. So in general, this is looking good. And again, in the plot TSNE, we can also activate the argument for showing the nearest network. So if we set this as true, we can add the nearest network here. So we are again observing the connections between the same cluster and their connections with the other clusters in our sample. And we can use again the spatial plot, but in the past we were coloring the cells depending on their number of features per cell or the number of for example, the expression, the total expression per cell. Now we can also observe the distribution of the clusters in the spatial context. So just to clarify, so far the calculation of the PC, the calculation of the UMAP, and the calculation of the network and the clustering is not taking into account the spatial information. It's only relying in the expression information, only in the expression levels of the genes. But tomorrow we will learn how to take into account the spatial information. So right now, what we can observe in this plot is, yes, the spatial distribution of the clusters, but take into account that this spatial distribution is just taken from the original location of each spot. So we are taking the original coordinates that were given to us in the data set, the X and Y coordinates of each spot. And then we are coloring that spot with the cluster that was calculated only based on the expression levels. Which is very nice, is that we can observe that even if we are not considering yet the spatial context, we are still able to see the different spatial layers in this sample. Now, when we take into account also the spatial context, we will get a nicer result of the different clusters. Right now, for example, we can see that some of this red layer is not super well defined. We have kind of a mixing a little bit with this other cluster over here. It's not super well separated from the gray one, but it's still giving us a good first result. So yeah, just wanted to clarify this, that so far we are not calculating anything based on the spatial context, but we have a way to do that. And we will learn that tomorrow. And As I was mentioning, the latent cluster is something that it's a method, it's a clustering method, very, very popular, especially when you don't have very large data sets, and with that you don't have very large networks. But if you have large networks, then probably you want to run the Louvain cluster. So the Louvain cluster will take a little bit more time to calculate because it will do kind of a repetitive calculation of the different groups across the data set. So it takes a little bit more computational power, but it's good when you have larger groups or like larger samples. So for running the clustering, you just have to call for this function, do Louvain cluster. The result again will be stored in the metadata. So if we take a look again at the metadata of our object, we can see that now we have another column. So we have the latent clustering, and now we have the Louvain clustering. And of course, these methods can give us a slightly different results. For example, here we are already seeing that we have 22, like an ID of the clustering with a 22, while in the latent clustering we have, I think, 16 or so clusters. Let me see. We have 19 clusters using latent cluster with that particular resolution. Remember that we can modify the number of clusters based on the resolution in the latent clustering. So for Louvain, we have 22 clusters now, and we can use the column name for plotting again everything. So we can plot again the UMAP, we can plot again the TSNE, the PCA, and we can plot the spatial plot using this column name. So let's see the result. So here, for example, we observe a more detailed construction on these layers. In the past, for example, we saw kind of a big layer over here, and then was just a very, very thin layer just in the out of the sample. The other clusters are pretty much the same in the big picture. We also have this line over here in the middle of the brain. So, of course, there is not like a perfect, let's see, workflow. What I recommend is just basically comparing the methods, see which one describes better your sample. And of course, there are many things that you can play with. For example, the resolution. So you can increase or decrease the number of clusters that you calculate each time. And we can plot again. I didn't add that here, but we can also plot the UMAP, for example, the UMAP from the Visium RAM, and color the cell. I will just copy the name from the previous instruction. So we can plot the UMAP using now the Louvain clustering. Let's observe the result. So here we have the different IDs again, and we also have the legend on the right with the different clusters. They look very well separated too. Let's look at the TSNE. So let's do again plot TSNE from the Visium brain object and using the cell color with the Louvain clustering. So here we have the TSNE. This is actually looking very nice. Here we have like the red. We have some yellow over here, the gray over here. So in this case, we are having really good results using both methods. And if you want to get more information about the comparison of the latent and Louvain clusters, cluster methods, sorry, you can look at this paper. I found it very, very useful. Just they explain in general how each method works and what are the main differences between them. So I added this to the book. And we are going to finish early today, actually. So tomorrow, as I was explaining, we are going to learn how to take into account the spatial context. So we will still need the first steps of the analysis for this object, which are the filtering, the normalization, and finding the highly variable features. So since we don't want to run everything again tomorrow, we can save our progress. So we can save the Giotto object. We have included this function, which is save Giotto. You have to provide as the first argument the Giotto object that you want to export. And then you need to provide the path to a directory that will be created. You don't need to create it before. You just need to provide at the end the name of the folder where you want to store this object. So this folder will be created. And then tomorrow we can load again this object so that you don't have to run everything again. We can load our object in the current state. So you basically have to just run this. In my case, it's saying that my folder already exists, so I can use the override argument equals to true. And with this, it will basically override my object. So it's going to start saving. This may take a few seconds, but it's not going to take too long. And I think with this, we will finish today's sessions. Do you have any questions about this pipeline for Visium technology? Okay, I think all the questions in the chat and the Q&A were answered. So if you don't have further questions with this, we are going to finish today. So my exporting of the Giotto object finished. If you are also following the code, you should see something like this. It's just saying that it started writing the feature information, in the spatial information, the image too, and at the end you should see this true. So with that, you should see in your folder, I saved it in results. So on the results, I have the Visium brain object. Since I set the override equals to true, then it's just adding the number 2 as the version number 2 of this object, but you shouldn't see this 2. You should just see Visium brain object.